When commercial poultry flocks experience catastrophic mortality from secondary infections, you are rarely looking at the fault of a single pathogen. You are witnessing the final stage of a prolonged immunological collapse. To diagnose how this collapse happens, we must separate two biological processes. The first is immunosuppression. This is a systemic, nonspecific dysfunction. A pathogen or stressor damages the host's lymphoid tissue, leaving the bird broadly vulnerable to almost any subsequent threat. The second process is immunoevasion. This is a targeted, pathogen-specific countermeasure. It exists strictly to protect the invading agent from localized host defenses. It is the difference between leveling the factory to stop production and cutting a single alarm wire to avoid detection. In a commercial setting, these two mechanisms do not occur in isolation. They execute sequentially across the life cycle of the bird, compounding the damage. This flowchart maps the 49-day lifespan of a commercial broiler. Look at the far right terminal block. By the time a flock is hit by infectious bronchitis or E. coli, the damage is already done. We must rewind to day 0. Controlling disease on the processing line requires tracking the exact timeline of stressors and viral saboteurs that systematically disarm the flock weeks before the first clinical symptoms appear. The degradation begins at hatch. The physical realities of transport, handling, and thermal inconsistency serve as the flock's first major stressors. These environmental variables trigger the hypothalamic pituitary adrenal axis, responding with a massive release of corticosterone. This acts immediately on the immune system, causing rapid depletion of lymphocytes and early physical atrophy of the thymus and the bursa. At the same time, the flock encounters its first dietary saboteurs. Early feed often contains trace fungal mycotoxins, specifically aflatoxin B1 and fumonisins. Once ingested, the liver bioactivates these mycotoxins, converting them into highly toxic epoxides. These epoxides bind directly to host DNA. They impair macrophage activity and actively downregulate the production of crucial early response cytokines, like interleukin-1 beta and interferon gamma. We can trace these inputs on the master model. The environmental and dietary insults feed directly into the timeline from the very first days of life. By day 14, the flock's baseline immune competency is artificially depressed. The primary viral saboteurs now have an opening. In the critical window of weeks 2 through 4, declining maternal immunity allows two specialized agents to enter, IBDV and CIAV. IBDV operates on an absolute timing rule. Infection before 14 days causes permanent immunosuppression, while later infections are generally transient. This permanence is dictated by cellular geography. Early IBDV specifically targets and destroys immature B cells residing in the bursa of Fabricius, killing them before they can migrate and seed the peripheral immune system. The virus uses a polyprotein called VP234 to clamp onto the host machinery, arresting B cell division without immediately killing it. Then, a second viral protein, VP5, accumulates in the plasma membrane, triggering the JNK pathway and forcing the B cell into apoptosis. With the B cells compromised, the second major saboteur strikes. CIAV infects the flock exactly as the last protective maternal antibodies wane. CIAV has a strict operational requirement. Its small genome means it must infect actively dividing cells, specifically hematocytoblasts in the bone marrow and T-cell precursors in the thymus, so it can hijack their DNA replication machinery. CIAV deploys a viral protein known as apoptin, or VP3, which enters the nucleus of the T-cell precursor and interacts with its supercoiled DNA. This induces immediate apoptosis. Consequently, populations of critical CD4-positive and CD8-positive T-cells plummet. By day 28, the systemic damage is profound. IBDV has dismantled the humoral B-cell defenses, and CIAV has wiped out the cell-mediated T-cell defenses. Later-stage pathogens now arrive in a devastated systemic environment. Because the host is compromised, these advanced agents rely on precise immunoevasion to guarantee their replication. Avian influenza provides a premier case study in this targeted evasion. Influenza produces a protein called NS1 that physically wraps around and hides the virus's double-stranded RNA from host sensors. Obscuring the RNA blocks activation of the PKR pathway. The cell is muted, unable to produce type 1 interferons to signal an attack. Marek's disease virus, or MDV, deploys a uniquely deceptive evasion mechanism. MDV secretes a protein called VIL8. This is an active mimic of a host cytokine. The false signal actively lures healthy, activated T cells directly towards the site of the infection. By using the host's own immune language to call T cells forward, the virus ensures a steady supply of target cells, establishing long-term latency. At the flock level, the timeline has reached a critical failure point. Advanced viruses have silenced the innate alarms, and the baseline immune system has already been gutted by IBDV and CIAV. The door is wide open. Opportunistic environmental bacteria like E. coli and parasites like coccidiosis sweep through the defenseless population. Immunoevasion keeps the specific virus alive, but it is the prior systemic suppression that dooms the host to terminal secondary infections. Zooming out, the entire journey from day 0 to day 49 presents a complete diagnostic picture. The ultimate biological and economic consequences sit at the very end of the timeline. The clinical respiratory distress or bacterial sepsis you observe on the processing line at day 40 is merely an echo. It is the delayed result of silent, subclinical immunosuppressive events that occurred at day 10. Immunoevasion is a pathogen's localized survival tactic. Immunosuppression is an existential threat to the host's entire life cycle. Effective veterinary pathology requires shifting your diagnostic focus to the left of the timeline. Attempting to vaccinate a 30-day-old flock that already has a decimated bursa and thymus is biologically futile. The machinery required to process the vaccine no longer exists. Mastering avian health means rigorously controlling early environmental stress and aggressively blocking those initial viral saboteurs before the cascade can even begin.